Good evening, everyone. Good evening. Welcome to Busy Bee, and for all of you at home, thank you also for joining us. We just watched a video of Carib Trans. Carib Trans is a company that does cargo shipping, but they also sponsor our live streaming. So everyone who is watching from home, please give a wide round of applause for Carib Trans. We'd also like to thank SATEL, which is the Saba telephone company, who is providing internet to all of our presentation venues so that everyone is able to see and hear our presentations. So a big round of applause for SATEL. As always, we'd like to thank our other sponsors. Our two main sponsors are Public Entity Saba and CCG, which is the former Prince Bernhard Culture Funds. Big round of applause. Yeah. Okay. And we'd also like to thank all of our other sponsors. We have close to 60 sponsors now. Many of them are in the audience. We have Andres from El Momo, Larry and Sherry from Flamboyant Cottage. We have Tim from Banana Kit House. We have Boomerang. We have Michael from Iris House. And I think that's all I see at the moment. But thank you to all of our sponsors. This event would not be possible without you. Is it working now? And a round of applause for Adam. We'd also like to give a big round of applause for the Cottage Club, who is sponsoring Alison and her crew this week. And as Lynn mentioned earlier, we have a raffle going on. They're $5 a ticket or a little bit of a discount if you buy in bulk. The drawing will be on October 27th, and you do not need to be present to win. So contact either Lynn or I and buy some raffle tickets. Upcoming events at a glance. Tomorrow, There will be a dive on the Saba Bank with tonight's presenter. We are told by Saba that it is sold out, but if you want to be put on the waiting list, let us know. And unfortunately, we have to cancel the presentation on Wednesday due to the effects from Hurricane Milton, which are going to hit Florida. A lot of our presenters are either live in Florida or traveling through Florida, so we are making the necessary adjustments. So Wednesday's presentation is canceled. And if you attend one of our field projects, you win one of these very nice pale mint colored sun shirts. So sign up for a field project and you get one of these really nice soft shirts made out of recycled plastic. And now the reason that you are all here tonight. Tonight's speaker presented during our 2022 program on Ciguatera, so we're starting off the evening with a little trivia. Does anyone remember what the most toxic fish our speaker's lab has tested for Ciguatera from 2 years ago? Does anyone remember? Not barracuda. What did you say? Parrotfish? No. Not lionfish. Think smaller. Not snapper. Not red hind. Small. Think small. Very small. Not sardines. No. Damselfish. Damselfish is the answer. So, Dr. Alison Robertson is a faculty member in the School of Marine and Environmental Sciences, University of South Alabama, and senior marine scientist at the Dauphin Island Sea Lab. Robertson is the graduate coordinator for a master's program in environmental toxicology and is the director of a large trans-ocean international partnership in Ciguatera research and education funded by the U.S. National Science Foundation. Dr. Robertson has been fascinated by organisms that produce toxins and venoms for over 20 years, working in a variety of different ecosystems all over the world. Robertson's research focuses on understanding the biological role and environmental drivers of natural marine toxins in marine microalgae and the mechanisms by which they exert their effects in the marine organisms. Welcome back, Alison.
I'm not sure this is working. Can everybody hear me? Okay. All right, good. I thought I'd have to get loud in here. Yeah, thank you for having me back. I really appreciate Lynn and Emily for all of their help and for hosting my team and I and setting everything up. And thanks also to the Cottage Club. It's a great place to stay. We love it. And to Sea Saba, who have been going out with us every day and looking out after us out on the water and all of our dives, which is quite a few and more to come. So I'm thrilled to be back. This is one of the most beautiful islands. I've traveled all over the world and I'm so happy to be back. And Liz was here with me last time and she was very excited when I mentioned that we might be coming back. So thanks for having us and thanks for coming. So let's get on with the show. This looks like a connection issue, but that's fine. Just roll in a whiteboard, we'll be right. Yeah. Sorry? Yeah, okay. Yeah, so we're going to talk a little bit about what's on your plate, thinking about seafood safety, but we're gonna go a little bit deeper into that and see what are the reasons for us being interested in what ends up in our seafood. So how does that work? What are the mechanisms? And hopefully that'll be interesting. So this is a picture we took just yesterday on one of our dives, and this was in Tent Reef Shallows. So for those of you that have been over there, you might recognize that. But of course, we've got a lot of biodiversity on these reef ecosystems, right? So there's a lot of different organisms, and you have some beautiful reefs here. But as we see changes in our climate and changes in the environment, increased temperatures in the water for too long, we can get corals bleaching and algae can grow over the top of those. Or we can have hurricane disturbance, right, that are going to cause disturbance and damage to the reefs as well. And some of these organisms capitalize on that. Before I go any further, right, this way. Okay, we're going to hold for a second. I'm going to introduce my team. So I have over here Liz Murphy, who has been with me for 5 years and came back to Sabre, so she's integral to this work. Ashley Delesk. You guys have got to make it a little bit louder over here because they're all helping with this research. Sarah Dietrich, who's a postdoctoral fellow. We've got Macy Knoll, who's an undergraduate joining us on this trip. We've got Dr. Sabrina Heiser and Mackenzie Pruitt and Kayla Hughes over there. And our always trusty volunteer, Ben, who's also my husband. He helps with all the other things. Did I get everybody? I think I got everybody. All right. But we got a whole bunch of people back at our labs and a lot of investigators and institutions that we have worked with for a long time in the US, but also overseas. So we thank them and their efforts as well. So why do we care about fish and fish toxins? Well, you're living here on a beautiful island surrounded by the water. So obviously seafood is an important part of life and livelihoods. And that matters for everybody that lives on the coast all over the world, right? And we have more coastal areas that are becoming warmer through time. So some of these issues are going to matter for that too. But every so often, some of these fish can become contaminated with toxins. And we think about toxins and pollutants all of the time, right? We know we don't want any nasty stuff in our seafood. We don't want them to be contaminated with microbes or something nasty and have a bad bellyache afterwards. But the toxins we're going to talk about have been around for as long as these algae have been around, so a very, very long time. They just happen to produce really cool bioactive chemicals that are natural. They are really cool, but it's unfortunate that sometimes they get into the reef ecosystem, so into the marine organisms, and can make those animals sick, as well as you, if you happen to eat one of those fish. So this has been, like I said, an issue throughout the Caribbean. This is a cartoon from a newspaper in 1980. So, and this is sort of the situation we're still in for Ciguatera. So if it's any consolation, we've taken it off the menu, right? So you can go to hospital and they will help you as best they can with the symptoms that you're having. But you've got to kind of wait it out. And this is something that when you're poisoned can last up to about 6 weeks. You know, typically 1 week or 2 weeks. But if you have a really severe case, you could be in intensive care and it could last 6 weeks. So it does matter, and we're doing what we can in my group and the collaborative teams from other universities to get to a better place of treatments and diagnostics and fish testing, onboard fish testing and such. So we'll talk a bit about that. And this data is also from a very old paper from '79, where they took over 3,000 cases of Ciguatera and sort of mapped out all of the symptomology and timing and severity. So I think I hit the wrong button now, but I'll try hit it again and see what happens. So we're good. I'm looking for the laser and I found it all on my own. Okay, yeah, even better. We'll go over here. So yeah, 1979. So here we've got days, and on the y-axis over here, this is degree of discomfort, 4 being the worst you've ever had in your life, right? You get those questions a lot, sometimes on a 1 to 10 scale. This is just a 1 to 4 scale. So this is pretty high, right? For within 6 to 12 hours, that's like, you really don't want to be dealing with that. So this is another reason to be aware of Ciguatera. With the gastrointestinal symptoms, can be seen within 4 hours and lasting up around 12. And this study, although it's from '79, these data still hold today when we look at the thousands of cases across regions. Myalgia, this is muscular pain on the top. So these neurological symptoms start kicking in after about 12 to 18 hours. Paresthesias and numbness and tingling and these sorts of effects, they start kicking in, but you'll see they sort of fall off a little bit, but they don't come down to zero, right? So these are the symptoms that can last quite a long time. And then a little later, after about 24 hours, you might get this pruritus, and this is a medical term for itching. So, and this is a neurological itching. So it's the kind of itch you can't scratch. So it's pretty awful. I don't know if you've ever had really itchy feet, and in the beds of your feet, that itch, that's the itch. So the only thing that seems to help is having, dunking yourself in a really ice bath. But that's all the rage these days. So maybe you want to do that. Anyway. Okay. So this phenomenon of ciguatera, or fish poisoning, is something that we used to consider really important throughout the tropical regions globally. So it's a global issue. Everyone is dealing with it in the same way throughout the Caribbean as they are throughout the Pacific Islands and the Indian Ocean. And we work in all of those areas for a reason, because we're trying to understand this phenomenon and the drivers. So what are the environmental conditions that are making this happen, and can we do anything about it? Can we predict what might be going on? But through time, with increased reporting, that's one of it, increased acknowledgement of this issue, and also with an increased prevalence, we're seeing an expansion of areas that are impacted by ciguatera. And then we have hotspots in other places in the world that you would never expect to see ciguatera cases, where we're having sometimes hundreds of cases of ciguatera, just through trade, right? Import-export of fish from really rich fisheries areas. So the tropics are really good for fisheries. It's a really nice area for fish. But if the wrong kind of fish are sold at markets in a region where people don't understand the fish because they're not local to their region, such as, some barracuda in New York City in 2010 and 2011, caused a lot of outbreaks, and that was just from a fish market in out of Brooklyn, and a lot of people were just purchasing all of that barracuda that was collected from a hot region, or a region where we know that ciguatera is prevalent. So the unsuspecting folks in these other areas end up with a nasty surprise in that case. But I want to say right off the bat, I'm an advocate for eating seafood. I love seafood. I plan to eat seafood while I'm here. And sustainable fisheries is very important to me. So I'm not here to tell you to be so worried that you're not going to eat seafood. It's just really good to be aware. And the more you know, the better it's going to be. You can, maybe avoid some of this illness. In terms of prevalence throughout the Caribbean Sea and Northern Gulf of Mexico, so the greater Caribbean region, as we would call it, we have quite a high prevalence, right? So the yellow is basically all of the coastlines where we've been working on outbreaks in different regions through time. And of course, that includes those here and throughout the Antigua Island chain, up through the Florida Keys, Florida coastlines, up into the Northern Gulf of Mexico, where we are. So we're based right over here. And our colleagues from Florida that are with us today are based right here, unfortunately, in the line of the hurricane. But they're here, so they'll be okay. But where do we find these toxins? What's going on here? Typically, the organisms that produce the toxins live here. These are the substrates of macroalgae and seagrasses. And these pictures were all taken off the coast here in Saba. And they live as tiny little microscopic organisms on the surface of all of this macroalgae. It's all this big algae that's growing all over the reefs. So we take this substrate and we shake them all off to then count and determine how many of those little toxic cells might be there. And that helps us understand how much of a toxin might be in a given area. But that doesn't mean that the fish are naturally going to all be toxic in that region, right? There's more to it than that. But these are all the things that we've been collecting here and that matter. So how do they live there? So the backbone or the thallus of these seaweeds and algae are this. So this is the substrate itself. And all of these little cells are benthic microalgae. So little microscopic algae minding their own business. They don't know that they're doing anything wrong. Right? They're photosynthetic organisms. They love light. That's what gives them their energy. And they live, and you might see on this one here, they sort of just hug the surface of these plants. So they're not really tightly bound to it. They're actually held on by a mucilage or some mucus that they embed themselves in, and they produce that mucus. And they can actually hang off this in tendrils when it's nice and calm. And they can even swim a little bit, but they're not really great swimmers. So you're typically not going to see them bloom or, flourishing in the water at all. They like to be on the bottom. And that's where they're happiest, but that's also where they're producing toxins. So what do they look up a little closer up? Well, this is in culture, these mucus that they're producing. They look like this. And this is actually a video. And they're kind of big compared to their cousins. But they don't really move around very much. But they remind us, when we're looking at them, of little bumper cars. Right? But they're these golden, beautiful little organisms. Harder to see is that they have flagella. They're called dinoflagellates because they have two flagella or tails that help them whip themselves through the water column a little bit. But they are really big, like they're too big to do a really good job of swimming, so they tend not to. My team here have a few little 3D printed ones that you can pass around and have a look at. But they're actually really beautiful on their surface. And they even painted them golden, so thank you to FGCU, Nick, for producing these so everybody could take a look. We're going to show these to the kids tomorrow as well. But although they look kind of harmless, right? They don't move around very much. They don't look very scary, but they are very good at producing toxins under the right conditions. Those toxins look a bit more scary, right? So you don't have to have done much chemistry to think, well, I don't like this already because there's too many carbons. There's too much going on, right? But these organisms are capable of producing these massive compounds, some of which are the largest small molecules known. They're also some of the most potent compounds that we know of. And they're all neurotoxins. So they affect your nervous system, which is why you end up having these neurological issues. Yeah? Yeah, the question is, how large are the dinoflagellates? They're actually between 60 and 80 micron. Putting that in perspective, you could think of it being a mouse and an elephant, maybe, in terms of, proportional sizes. Yeah. So, yeah, but they're big for who they are. So their neighbours might be 10 times smaller than them. Yeah, but they're pretty big and they take up their real estate. So can you think of what would be the purpose? So these are really big compounds, right? I love this stuff, but I don't expect everybody else to love the chemistry. But if you were, this takes a lot of energy just to look at. So to produce them, like, why do you think they might produce them? That's a good, somebody said it might be a defense mechanism. That's a great hypothesis. You're on the team. Who else? Any other thoughts? Why could they use these toxins? Yeah, to survive. They could be some kind of energy storage or something. Yeah, maybe. Yeah. You can also come on the team. Any other hypotheses out there of why they might produce these toxins? Maybe. Yeah, it might help degrade something. No idea? Yeah? Maybe. Yeah, maybe. Oh, I don't think they even know we exist. Sorry? Oh, possibly. They actually use light for their energy source, but it might help them with light harvesting and something like that, utilizing that. Anyone else? Any other thoughts? Love it. Yes, definitely on the team. Yeah, sorry, the answer was for competition, and we had, some more chemical defenses and helping them to use energy and so on. All great hypotheses, and honestly, I wish I had the answer for you. But now, welcome to science. It's a brutal world where we don't know the answers, so we're constantly trying to find them out. But great hypotheses are that there might be anti-grazing deterrents, right? So trying to ward off high levels of grazing. That's one of them, except fish and invertebrates are still grazing them and becoming toxic. So that's not working so well. Other hypotheses have included, some kind of carbon storage. So each of these are big carbon rings, so it might be a way for them to hang on and pack some carbon into their cells because they need that. But the biological role of these super bioactive toxins are a little bit, we don't know. So I find it fascinating and I really want to find out. So unfortunately it's been 20 years and I was still trying to find it out. But, perseverance, right? That's what we're going to do. But we're going to get to a little bit of it, right? So we have lots of questions along the way that we have. So there are many different species of these Gambierdiscus, so these toxic cells, or potentially toxic cells, but they don't all produce the same amount of toxins, and some of them are completely harmless. And in some regions, they can produce toxins, and in others, they don't. So thanks for that, Gambierdiscus, making it tricky. But this figure here, I'm showing you 4 different species that are really common, and we found them on the algae that we collected here in 2022. And this is a log scale. Don't worry about it too much. It just helps you look across 10 orders of magnitude as you go up the scale. That just means that these ones compared to this one are very different, but I needed to have this scale so that you could see this on the same graph. This would go up to the sky otherwise by comparison to these if I had them on a regular scale. So these ones here are up to 1,000 times more toxic than the other ones, unless you're in the Florida Keys, where the isolates that we've tested there are non-toxic. So interesting, but that gives us something, right? We can then compare and contrast the environmental conditions, the substrates they live on, and we can start asking different regions, like what is different about these two places? Yes? You may have missed what is LOD? Sorry, yeah. Level of detection. So these is really, this is the limits of our methods at the moment to detect the toxins. So everything above that, or below that, is sort of trace. We can sort of detect it, but from this point on, it's a reliable detection. So we're really confident in those numbers. And then everything above that is really quantify it. Like we can quantify it. It's really robust measurement. So there are differences among species. So this means, one of the most abundant species on that list is actually this one, so Carolinianus, but it's, one of the lowest on our list here. Whereas, so if you compared those two in very simple ways, you could have Carolinianus present, and 1,000 of them, and that would be equal to 1 of the Gambierdiscus sylvae, right? So we used to think, and the standard thing we do in the study of harmful algae, so all of these algae that produce toxins, is to count them, because we think that the number should correlate to the amount of toxin. This kind of blows that theory out of the water, so we have to rethink our strategy for monitoring and management of these algae in these benthic environments, or those that live on the bottom. So we're working on that as well. And then there are lots of different things that might matter. So we have all of these are just representative macroalgae here, so Halimeda and Dictyota and turf algae, which are just the little fuzzy short algae that live all over the rocks and other things. Fish actually really prefer to be feeding on that turf algae. But this is really incredibly difficult to sample effectively and in a way that we can measure in the same way every time. So what could we be missing here? Do they prefer to live here versus here or on this other nice green Halimeda? And then we have issues of, as the temperature changes, each one of these species has a different preference. The Gambrodiscus sylvae, this toxin producer, at least in the Caribbean, the lower Caribbean and mid-Caribbean, prefers to have cooler temperatures than all of the other species. It doesn't like as much light. So right now, these hot water at 30 degrees would be pretty uncomfortable for these toxin producers. So that can be some good news, even though it's like bad news for other things. But then we can have wave action and turbulence. Remember, these cells like to cling to these surfaces on the bottom. And if you're diving when there's a lot of turbulence and surge, we were at Green Island the other day, and there was lots of surge. And you can see all of the material like sloughing off the algae into the reef. So the cells are then being disturbed, and they don't like that. They want a nice, calm, settled kind of environment. So wave action can make a difference, and light can make a difference, and light can be very stressful. So that actually means that some of those toxic species prefer to be deeper in the water column than you would expect them to be, which is why we're at the Sabre Banks today, sampling some of those deeper sites. But we have our collaborators at Woods Hole Oceanographic. So this is a PhD student, Katie Pitts, with Mindy Richland and colleagues, developed some cool tools to start determining the difference very quickly of these Gambrodiscus that live on the surface of these macroalgae using these fluorescent probes. So we call them fish probes, but that means fluorescent in situ hybridization. It's a little piece of DNA that's recognized and it glows. So it has these nice color differences depending on the species. So each one represents a different species. This usually shows up a little bit more orange here in this panel here. But then we can get a better idea of counting them and who's present, because we can do it by species instead of just doing everybody that looks like a brown cell under a microscope. So that's some things that have helped us then determine the prevalence and distribution of all of these different types of cells. So here we have a plot of the relative abundance of the different types of Gambrodiscus in the Florida Keys compared to St. Thomas, so a little bit north of here. And we've got the Belizeanus, Carabeus, and so on. The dark blue sylvae is the dark blue is the sylvae, that toxin producer. And you'll see that in St. Thomas, there's more of that. And this is also a hyperendemic region for Ciguatera. They deal with this a lot around St. Thomas and St. Croix and St. John. But in the Keys over here is really only one site where you see that sylvae, right? So this distribution helps us, and it matters. Yes?
Could you back up one slide?
Of course.
Wouldn't the color be grossly affected by depth?
These colors are actually once we've collected the sample already. So we bring them up from the bottom, and we mix them up, and we look at them under a microscope.
So this is in the lab.
Yeah, this is when we get back in the lab. We can treat them with a few chemicals while they're still living, and then they'll glow when we hit them with a bit of UV light. Yeah, good question. But the other thing we always want to remember, and this matters for the data that we've collected here from Sabre, is that the Gambrodiscus, although there are many species of them, they're not alone either. There are other types, other cousins and other dinoflagellates living in the same environment, and they seem to be, we think, happily living together because we find them together all of the time. So this is a microscopy, so it's something we look at under the microscope after we've pulled the seagrass up, shaken them all off, and just put them in a little slide under the microscope. And you'll see These big orange cells here are Gambrodiscus. This one's on its side, so they're all rotating in different ways, right? But then we've got lots of other things here. So we have these little cells that look like little pears. Those are another cell called proricentrum, which can produce other toxins. We have these little orange cells, these ones. These are actually called coolia, and they also produce very potent neurotoxins. So we're working on all of them. So do you think fish can recognize which one is which? Do you think they're choosing the big cell over the little cell? Probably not, right? So they're probably consuming all of these if they're feeding on that substrate. So if they're eating the seagrass, they're probably going to eat all of these at the same time. So unfortunately, very rarely has the community of scientists looked at them all together. But there's been a shift and a change, and we're happy to say we're now thinking of them as a collection of algae, like this soup, toxic soup that they might be producing. And this is what that suite then starts looking like if you include those other species. So a bit more diversity across those. So it matters. So I don't expect you to remember these structures, just to say that they're quite different, quite diverse, and there's quite a few of them. So if we think about this in a reef ecosystem, we have the Gambrodiscus here, microscopic, living on this Dictyota here, you know, some fish feeding on those, and then those fish are being fed on by others. These invertebrate grazers are also potentially eating the same stuff, and all of them can produce toxins and become toxic. So you would think that would mean that this would work like mercury, where you get biomagnification, where these top predators, like the groupers and snappers and barracudas, become extremely toxic in these environments. But what we actually find is that there's a weak evidence of that biomagnification. And these toxins from the algae through the grazers or browsers like the tang, all the way up to these higher predators, are really in the same order of magnitude. So what's going on there? They're actually converting those toxins as they go into different things. So the toxins that are in these apex predators are a little bit different than those that are produced by the algae. So they're being changed as the fish and other invertebrates are digesting them, right, metabolizing those things. So this is halftime. So if you have any questions at this point, I'm happy to take some. Yes? Well, yeah, I mean, the diadema die-off, I don't know. Sorry, the question was, would the diadema die-off have, so sea urchin die-off, have any effect on any of this issue? And the answer is it's possible that it does. So I don't have the answer except to say that urchins are very good grazers and they can keep some of this macroalgae in check. So what you've done is basically populated the real estate market out there for Gambrodiscus, so there's more space for them to live. If so it's a very good point. Yes, I'll come back to you. Haven't forgotten you.
A given level or given species of Gambrodiscus, are they uniformly toxic, or are there perhaps some environmental stressors that raise or lower the level of toxicity?
Fantastic question. We could be friends. So the question is, within the species, different species of Gambrodiscus, do environmental parameters alter their toxin production? Yeah. And the answer is absolutely, yeah. And we're trying to understand what that is. So back at the lab, both in Florida and formerly in Texas as well, and in Alabama, we have 500 or 600 of these organisms living in the lab in culture. And we're doing experiments to try and understand how they are impacted and how the toxin production changes with those different factors. It's a harder thing to measure in the field because everything's changing so much and it's hard to capture them. But really good question. But yes, they all they can be very different depending on those environmental conditions. So my collaborator Mike Parsons is doing a really nice job trying to develop some predictive models based on those experimental things. I'm going to go over here and come back to you. Yes, next question.
So with the echinoderms here on Saba, I know there's a real problem with them going out and grazing and dying. Is this algae part of their grazing?
It would be, yeah. The question was, are the echinoderms that are present on the reefs here in Saba that are apparently dying off a little bit, could this be a reason why? I'm not sure, and my answer is I'm not sure if it is or not, but they are certainly grazing on a lot of these other substrates. Not all of them, so more what we'd need to really do a study on that. So.
Yeah, and down at the Saba Conservation Foundation, they're doing a lot of work with this
Yeah.
On inside tanks.
Mhm.
So
Yeah, it could be a great thing to test. Yeah, I love it. Coming back over here. Yes, sir.
Two little questions.
Yeah.
You had a map with Florida Keys with pie charts, the relative abundance of the species. Is that a snapshot, or is that, is that sample one event, or is there seasonal aspects here?
Yeah, good question. Yeah, the question was about the pie charts that we showed earlier. Doop, 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 doop. And if they included, next one, did I go too far? If they included a snapshot of the community or if they included some seasonality. And here we actually have hundreds of samples that were collected, and these were collected monthly. So it's somewhat of a composite. Yeah, it's sort of what happened across those. So we do have other plots and data that we could show that I haven't got today, but showing the seasonality of those changes. And we do see significant seasonality in toxin production, where toxin production is higher in the cooler months. So that data from the Caribbean anyway, from St. Thomas. Okay, another question, I love it.
Are there any latitudinal shifts in the abundance of, or the incidence of toxins in the Gambler discus arctic?
The question.
Water temperatures change latitudinally
Yep.
with climate change.
Yep.
Point.
Yep.
We're seeing shifts in plant species northward
Mhm.
from the plants.
Right.
So do we see anything like that?
Yeah, good question again. The question was, do we see a northward shift or a transition as we get towards cooler waters? And with climate change, all of the waters are changing, right, and becoming warmer. So we're getting tropicalization, and species that used to be only in tropical areas are now in warm temperate areas and temperate areas. And do we see a change in Gambler discus community with that shift? And yes, we do. We do see a difference, but we don't have all the answers yet on how it matters. Correct. Because it's quite intensive to sample these because you have to be on scuba, getting them quite carefully. But with enough people working on this and we're growing the scientists, it's not. No. I'm less interested in the cold water diving, but it's important research. Yeah.
Has anyone looked at medical applications of these toxins in lower dosages, like toxins?
They have looked at these and many other toxins like them. The one thing that we'll say about these structures is they're quite complex and quite big, and they get in the way. So they cause interference with other mechanisms and things. So it's anticipated in the little work that has been done on medical applications, which is the question. They cause too many other symptoms because they're so big. But people are trying to chop it up into smaller pieces and see if some of the components can be used. So there are other neurotoxins. One of them is called saxitoxin. It's produced by, we see that in other toxic species. It's being used as a local anesthetic in surgeries like in knee surgery and everything. You can use it as a nerve block. So, yeah. Good question. One more question. Yes.
You kind of went into symptoms of us as like consuming
Yep.
as humans. Like
Yep.
what is the experience or symptoms for the fish?
Great question that I'm going to bring up in the next part. Yeah. I love it. That's a great segue. So thank you for the questions. I like to have half time so that you don't have to remember them all. Let's go. All right, so we've learned, this is the little recap, that there's a high variation in substrate, and these different types of benthic microalgae, the microscopic guys, can live on different ones, and they might have a different selection, right? They might prefer to live in one place versus the other. These are things we're trying to understand. And this is what makes it a little bit of a complex story. Then we see variability in the types of these benthic dinoflagellates that are present, and they can vary in the toxins they produce and the metabolites they produce, and in the toxicity of those metabolites. So that makes it complex. Then you can have a variety of primary and secondary consumers. So you can have everything from fish grazers and like parrotfish and browsers like tang, to gastropods that are actively feeding, and other echinoderms and urchins and so on. Then you might have a variety. Now we're getting mobile, right? Things are getting even more complicated because once things start moving, it's harder to track. But you have these tertiary consumers with varied diets and varied life histories. Some short-lived fish, some much longer-lived fish, hence the damselfish being one of the most toxic fish that we've seen. It was also a 40-year-old damselfish that was aged and very, very toxic. But some of these fish have, a tendency to live close to home, and their range is quite small. And because of that, they're living in an area, but, if that area had a lot of these toxins, then they have a higher potential through their lifetime to become more toxic. Versus the complex, even more complex and harder to predict story here, of very mobile vectors. In snappers, if we went to mackerel and others, highly migratory species, that can be even harder to predict because they're moving great distances between regions. Something like a barracuda, we do get poisoning events in the northern Gulf of Mexico from barracuda that people have consumed, but it could have come from the Caribbean, because they are able to travel 1,200 to 1,400 miles based on the telemetry work or the tagging studies that have been done on those fish. So it makes it really tricky. But we're not giving up, even though there's more questions, on how those toxins change. We call that biotransformation, so how they're transformed into the next thing. If and how they are actually biomagnified in some species, and how toxic each of these organisms become. Each time and at each step throughout these pathways, those toxins are being modified. So it makes it a very big chemical ecology story where we're trying to understand those chemical changes and how that might affect their behavior, their reproduction, and just in general, their fitness, right? How well they're doing on the reef. So let's go to Sabre. We were here and then after this, I'm coming back to behavior, I promise. We sampled at a number of sites. So we were only able on our last trip to do a few spots, and it was quite windy and the temperature was much cooler than it is right now. But we sampled at Hole in the Corner, over here, Tent Reef Shallow, Customs House, over at the Twilight Zone, with some visiting scientists that were also here speaking as part of See and Learn. So they came and did some of those deep dives. Up at Torrens Point and Man of War Shoal. And we were collecting those nice, as many different types of those macroalgae, so those big substrate kind of algae, as we could, and we took them to the lab to work on them. We also had the part of the field activity, we had a bunch of people come with us to Man of War Shoals, and that data is included in what I'm going to show. I guess I don't have a graph because I tried to make it a little bit easier on the eye. But those data are real, and we're using those data. So those samples turned out fantastic. So if you want to come out, we can find a way to get you out, hopefully, maybe, to come and help us with the sampling. It'll really help us understand what's going on in this area. So what do we do underwater? We have here Rachel and Kaylee, who were here in 2022 with us, collecting algae, and this is just a simple hard-sided Ziploc container that you might have in your kitchen, like a Tupperware, and they're putting some of the substrate here into this container, cap it up, and we come back up. Well, we collect lots of them, and then we come back up. And this is Liz, who's sitting over here. This is in the Florida Keys at the Keys Marine Lab. But with all of these algae that we've been diving and collecting, and she's been shaking them up and now subsampling different things from them so that we can collect a lot of information. I was trying to find a really good video of that, but there was a bit too much going on. So we take this sample. This is a little flow chart here. We have this, we collect the samples underwater, we shake them up 5 times, and we put them through this like really fine mesh so that we can catch all the little guys. And once we have those little guys, in here, we shake them, they all end up in this tube over here, like all in a suspension. So now it looks brown because it's got lots of little epiphytes, so these little cells in that tube. And then we put them into lots of different sampling efforts. One of them is to grow it in the lab, which we call a live sample. That way we can experiment further on them because we can grow them up. And a second one of them we preserve to look at them under a microscope and count them. A third one we preserve in a slightly different way so that we can put those fluorescent probes on them and count all of the species. Then we do metabarcoding, which is a way for us to look at all of the community, so every possible type of organism that's in that sample. And then we have another pellet that's left for us to look at the toxin production. And then we use all of these data from these other samples to help us understand what that toxin, production means per cell, per species, and so on. And then we keep that macroalgal sample so we can make sure that we determine that it was the right species. In different regions, things can look different. So what did we find? Getting to the point. Across all of these sites, including the field activity, we had toxin, really a lot of bioactivity happening from the samples that were collected over in Man of War Shoals and Torrens Point. So that's really interesting to us, so we're going to sample there again. That does not mean, so this is not a seafood safety warning at all. This is just saying that it's an area of further investigation because we sampled at one point in time, at 1 week, right, that we happen to be here on Sabre. So things could look very different through time. And that might just have been one little peak, right, that happened. And one little peak isn't going to necessarily mean that we have toxins moving into the food web. But we're going to do some more work, and we're going to try and see if there's a trend. So this time, we are sampling, just so you all know why we're here and what we're doing. Here's a little map taken from Littler et al. So this was a study done on the macroalgal diversity here on the Sabre Banks. And they show here Sabre Island, right? And the Sabre Banks, of course, is off the coast a little more. And this area here is known as the Poison Bank. And all of the surrounding islands that we work on, so Antigua, St. Kitts and Nevis, St. Lucia, and others, believe that that poison bank is an initiation point or a hot spot for ciguatera poisoning, so for this fish poisoning. So fishermen from surrounding islands actually avoid that area for fishing. So we thought it would be interesting to go take a look and see if that correlates to what's on the bottom. So the team were out there today and collecting samples, so we'll report back on that once we have the data, because it's an interesting one to test. And then tomorrow we hope to go out to these sites, Browntown and Seaweed City, and that sounds fascinating to us, and see what's there. And we want to do that because we have found, this is from St. Thomas, when we look at different fish species, so these are the queen triggerfish. Queen triggerfish, we've got red hind, the white grunt, and yellowtail snapper. That we see very big differences depending on where we collect those fish. All of these fish having a pretty small home range, so they don't go too far. But we found that on the leeward side of the islands, of course, were the hottest sites, but particularly offshore in the south. And in the north, none of those fish were toxic. So this is just good information, right, where you can work with fishermen. And this study was done completely in collaboration with fishermen in a co-production kind of way, where they helped us with the collection, sampling, and everything in between. So it was really great to collaborate with them. And we do that sort of work all over the world, so it's better to do it with everybody involved. Whereas, when we start looking at the more mobile vectors, like barracuda and mackerels, like Cerro and kingfish, it's a bit harder. So we start seeing different levels of toxin production. Here are on a risk rating that I put from 0 to 4. Green is non-detect, and then 4 is like a really high level that would make somebody sick. And you'll see it's a little bit more sporadic. So up in the north, where things were quite good for those other species, we've got green and orange and red. So it's a bit harder to predict in those species. But we're trying to develop tools and methods to detect the toxins more easily, so that fishermen can have a quick test on board and be able to hopefully do that without killing the fish and throw them back. That's the goal. We're still a few years away from getting that working properly, but we're definitely working on it, along with a lot of other people around the world. So toxin detection in some places might look like this, but I had an intern this summer working on this, and we've had past undergraduate students and high school students working on these traditional methods for detection. But at least in our hands, we couldn't reliably detect a toxic fish sample using these strategies. But we do use very high-end instrumentation to do that very well. But unfortunately, that's not easy technology to haul around with us in a backpack. So yeah, we're trying to find methods that are quicker and easier. And some of the questions we have, this is just a schematic talking about the main parts of our research, which are trying to understand how these cells produce the toxins, when, where, and why, and under what environmental conditions, and then understanding how they move into fisheries species that are important. And we try and understand how do they get into the fish, so how easily the toxins get in, when they're in the fish, which part of the fish are they deposited in one tissue versus the other, because if we could just remove one part, could we eat that fish, you know? Then we try and look at the metabolism, so how they're modifying these toxins in their tissues, and how they're eliminating them. So one of my students, Clay Bennett, has worked on this for his PhD. He was trying to answer, this was his hypothesis or his question. In experimental conditions, he fed fish, in this case a small reef invertebrate, ciguatoxins for a period of 3 weeks and watched the toxin levels rise, right? So these fish became toxic because he actively fed them toxin every day at a sublethal level. And we then wanted to find out how long does it take them to clear the toxins from their flesh, right? A really important question for fishermen, something we're asked all the time. If there's a pulse of toxin production in the algae, you know, that might then move into the fish, but how quickly could it come back down? So this was the question, and we thought that it would start to come down, right? Unfortunately, in this species that we tested, it didn't do what we thought, and it took a very long time, and that a half-life was actually 90 to 150 days. Experimentally, it was 94 days at a half-life and modeled out to about 150 days. So we really have to work on this and see is there something we can do to help with clearance from the fish, you know, because in the wild, there might be other processes at play that would help that we're missing in our experimental design. So more on that to come. But a really good question that came at halftime was, what are the effects of these toxins on fish? Because they do affect the fish. A common misconception was that the toxins had no effect at all on fish. But they very much do if you watch them closely enough. On the reef, it's very difficult to look at fish behavior, actually, unless it's really wildly obvious. Because we're moving and they're moving and lot is happening. But this is a fish, this is actually an arena. So this is a fish tank, a round fish tank, where we have cameras underneath and we look at fish swimming behavior, right? And this is a heat map showing where the fish swam. And you can see this fish went all around this tank, right? When we look at a fish that has been fed a diet of ciguatoxins for a long period of time, it looks more like this, where they hug the sides of the vessel and they don't venture in and out. What do you think that would look like on a reef if this behavior was happening on a reef? Huh? Maybe. Yeah, they might leave. Or hug the reef. Yeah. And they might look a bit strange because they're usually happily moving between places. Maybe then they can't get to food sources. There's all sorts of questions that come along with that, right? All of which need to be tested. I don't have all the answers to all of those things. But then if we look at these same arena, so these are small fish. This is a model species. And this is actually a freshwater species, but don't shoot me. It's a really good model for the neurotoxins, and we've repeated all of this in a lot of reef and marine species. But we're going to show you the control swimming along happily, and you're going to see a light over here that triggers a stimulus event, and it's actually a mouse trap on the side that snaps. So now you see it's a startle response, but in a control fish, it goes back to swimming normally after a while. This one here, the stimulus hasn't even started. After 30 days of a low-level repeated exposure, when that stimulus has started, I don't know, can anyone see a difference there between those two? It's crazy, right? Yeah, it's significantly different. So what do you think this might mean in a reef if you have a very hyperactive fish? Yeah, it's going to get eaten for sure. What was? Yeah, definitely too much toxin in it. Yeah, they definitely can. These fish don't do that, but yeah, in some species you could see that happen. When we do this, we have lots of different types of tanks and types of situations and different sizes, and we have lots of other ways of testing their behavior, but this is just the quick startle response, which is a really standard neurological test for these fish. But there's loads of different ways that could and should and have been tested. We see lots of differences. We haven't been testing that interference, but we have students that are actively asking those questions next. Yeah, what it does do, so this definitely depletes their storage. So this is 28 days, and once we get to 35, 36 days, they've actually, we have to euthanize them, so we have to kill them because they've used up all of their body stores, right, because they're so hyperactive. So, yep. But this brings me to other behavioral effects. So we've been working a lot in Florida, and you may have heard of some of the spinning fish behavior that's been happening and sawfish mortalities, which we've been working with the Bonefish Tarpon Trust and a lot of the Lower Keys Guides Association, Florida Gulf Coast University, and Florida Fish and Wildlife Commission. And during this time, we saw really strange behavior in fish. This is a video I took of a fish that was pulled out of the water. It was behaving this way in the water, but we put it in a live well. And swimming upside down, and we see a loss of equilibrium in those tanks. And although you see this very randomly from time to time when you do put fish in a live well, this is significant, and the fish that are being caught are doing this on the line, off the line, near the boat, and so on. But we saw behaviors like not just spinning, which might have been reported, but there was a loss of equilibrium, erratic swimming, so spiral swimming as well as this more circular swimming, vertical breaching, a reduced fight or flight response, so less fight on a line, and impaired gill movement, and they become very docile. So for a species like a jack, which is a very highly active, strong swimming species, it was the first time I was able to very calmly hold it in my hand in the water, which is very strange. Anyway, all of this being exacerbated by other additional stressors, such as light and all sorts of other things. Really strange things going on. But we found in the water column at the same time increased levels of these benthic algae in the water and increased levels of the toxins that we've just been talking about tonight. So we're doing a lot of work now to try and understand those effects and how they work, and if those alone cause these effects, or if other factors matter in addition to that. So this is that sort of spinning behavior of the snapper, and it is quite unusual in these species, and working with all of the fishermen and guides that are with these species all day, every day. They know the fish behavior better than anybody. So to them, this is a very strange thing, and not common. Sorry? Sawfish were also thrashing, yeah, and having... They're in the similar region, so the sawfish mortalities were happening actually very close to all of this. So yeah. So we think it's related, but we are handling the sawfish mortality event. We're doing them all at the same time, but somewhat separately. And we're not assuming one is the same as the other. But here, I thought it was important to talk about these. In the lab, we have grey snapper, where we're doing a variety of tests to expose them directly with the benthic algae and see if we can mimic these behavioral effects. So.
Fish like this survive in the normal water?
They do. That's what's been really great. That's the good news associated with that. Oh, I will repeat the question. I'm really sorry. Thank you. The question was, do the fish, have they been able to go back to normal after this? And yes. So the fishermen were putting them into fresh water, and within 30 minutes, 40 minutes, they had recovered and they could release them. In the lab as well, the exposures that we have done, we would see the behavior happen within 20 or 30 minutes. And we put them in fresh water, and they would recover in about the same amount of time, 30 minutes, 40 minutes. So, fresh seawater. Thank you for the clarification. Yes, no, I didn't stress them out even further by that. Clean water, we'll call it that. The question was, did I put them in fresh water? No, I did not. I put them in fresh seawater, clean seawater. So this was just to thank everybody that's helped fund some of the work we do. And of course, I've only shown you 1% of all of the things we're doing. But we wanted to thank you for your attention. And I thought I would leave it on this public activity that we did last year here in Sabre, with these great recreational divers helping us out with sampling. But if you have questions, we can talk about that. Questions?
Too early to know anything about the effect land water is having and the toxins, I mean, the stuff coming off the sides on the fish. Because we've had so much rain, there's been such a lot of fresh water, rainwater, bringing stuff down into the seabed.
That's a good question.
I presume somebody will be looking at it somewhere.
Yeah, so the question is, have we been, could we be, is somebody looking at the effect of freshwater runoff from all of the rain, pushing everything from land and off the mountain into these coastal ecosystems? And is that affecting the fish? And I don't know if somebody's working on that here, but yes, people are working on that in lots of different places, and I assume they also would be looking at that here.
Question. Have you raised the water temperature so high that it's actually killed the toxins? I mean,
Good question. No, good question. Yeah, so the question was, have we raised the water temperature to the point where the toxins start breaking apart or getting rid of them or destroying them? And unfortunately, the toxins that I'm talking about are extremely stable. So we can put them up to 120 degrees Celsius, and they are still present. Yeah, they're okay. But that doesn't mean that, they might attach to the sediment or be buried or, yeah. But it's a good question. Just behind you, and then I'll come back to you. Yes?
For swimmers that take in some salt water that's contaminated.
Good question. The question was, what does it mean for swimmers if they're swimming in an area where there might be these algae and potentially toxins. We've actually had no reports ever of any recreational swimmers having any negative effects associated with them. And that might be because the levels are too dilute, in a large body of water, which is the good news. So you don't have to worry about swallowing the water if these cells are detected. To our knowledge, don't sue me if that changes. I've just got one here, and I'll come back to you. Yes, sir.
Less of a question, more of a conjecture out loud, I guess.
Sure, yeah.
So at their worst, they're probably not interested in bait on a hook.
Right.
So not likely
Yeah.
to end up on a dinner plate.
Right. Yeah, so there's a comment, maybe because they're, when they have these toxins, maybe they're not interested in bait or getting onto the hook that you're trying to catch them with. And yeah, that's a really good thought. Yeah, I'm not sure. How would we test that? I feel like we need to ask the fisheries scientist in the room how we would test that too. But yeah, it's a good thought. That might be the case. They certainly don't fight on a line when people are trying to catch them, but I like it. Now you've got me thinking. All right, yes,
Okay,
another question.
So what's the conclusion? How do we make an informed and sustainable decision when we walk into a restaurant?
Well, you should know where your fish have been caught, right? So it's always a really good question to ask. The question was, how can you make an informed choice when you walk into a restaurant on what fish you might eat and what you might choose? So when you walk into a restaurant, what I would do, what I do when I go to a restaurant, so you can choose to take my advice or not, would be to find out where the fish were caught and what kind of fish, you should know what type of fish you're eating. And if that's a reef-associated species that lives close to home, doesn't have a very big, range, then, and if you know that there's a lot of toxin in that area, you might avoid that fish. That's a lot of information though, and that information isn't necessarily known everywhere. But there are some better decisions you can make, and like avoiding some of these apex predatory fish that like barracudas and mackerels that we know can, become quite toxic and are highly mobile. So, yeah. No, you should still eat fish. Fish are great. I'm going to come back over there in a second. Yep, one more question.
What is the correlation between healthy coral reef and bleached or bleaching coral reef and the upcoming of the species?
Yeah, good question. The question was, what is the correlation between damaged or bleached coral reef and the prevalence of all of these algae and these species? Great question. Even 50 years ago, people were asking that question when things were... I forget about there's cameras everywhere. I've got to pay attention to where I am and what I'm saying. Degradation on reefs, does this matter? Well, we do see people have been asking the question for 50 years if that has an impact. And we do see that macroalgae are the winners in these competitions. So those that are providing the host, are growing really well under those environments. In high temperatures, they can tolerate that quite well. In general, there are some species that don't, of course, which Sabrina Heiser over there could talk about. But so we think because there's more of that host algae present, then there is a hypothesis that could be causing an increase in different regions. More data is needed, though, I think, because it's very rare that all of the things are being asked at the same time, because the effect of degradation, right, is complicated already. And then layering these things on, I think we've just got lots of exacerbating effects that become intertwined, and it's hard to tease apart if it's one or the other. Yeah, the question was, in the Great Barrier Reef in Australia, is there any data that shows changes with changing coral reefs and temperatures and so on? And yeah, there's a lot of data in Australia. Over the last 20, 25 years, there's been a big surge in research there that's been done. Most of the primary research has happened in Australia and been developed from there. So they are drawing some conclusions about degradation and bleaching and proliferation of these species. Had the question over here. Yes.
I noticed when you were showing the Virgin Islands with the different toxicity levels and the different types of fish, I think all but one of the red fish was barracuda. Any comments on why? Or
Actually, a lot of them were different species. So the cerro mackerel and the king mackerel and the barracuda, but they're all long-looking fish. So my little symbols, it's kind of hard to draw them in a different way. Yeah, yeah. A lot of barracuda, yeah, for sure. But there's quite a few. Yeah, it's actually quite mixed. So the question was about the graph or the schematic or the picture I had with the different risk rankings of the apex mobile predators like barracuda and king mackerel, and if it was just mostly barracuda or if the others also had toxins. But yeah, they were definitely present in all of those species. So, another question?
People I've known who've been infected with ciguatoxin say that once they're over it, if they eat infected fish again, nobody gets symptoms but them.
Right.
Is that true? Has that been confirmed, or is that just?
Yeah, it's a great question. The question was that in people that have had ciguatera before, so one time, two times, that they might be more sensitive to the effects if they ate a fish the next time, that they might be the only one that got sick when others may not. And yes, there's a lot of reports of that, like significant reports of that, that people become more sensitive to the effects of the toxins with time and not more resistant. And that's different to other toxin types and classes where there are mechanisms that are upregulated, like things that happen, right, like antibody production and other things to help alleviate the effects. There are some studies that have been done in mice and rats, because this is where people start, right, because you can't work on humans, where they did some gene expression studies to look at what genes were up and down regulated in response and to ciguatoxin exposure. And they found that a lot of genes that are associated with the autoimmune response are upregulated, and that's thought to be linked to the sensitization in people, so following acute response. And there are some people I've talked to through time, because I've worked on hundreds of outbreaks and talking with people one-on-one and with doctors and other clinicians. And some of them have had ciguatera 20 times, and they live on the coast and that's their protein source, so they continue, but they can be hit very, very quickly, whereas somebody coming from the north isn't affected at all. But then if the same thing happens, if they eat a fish again, then they become more sensitive too. So thank you for bringing that up, though. It's a great question and a great thing to think about. Well, thank you all. Appreciate your time.
Alison.
You're welcome.
We invite you all to stay for dinner at Busy Bee. They have chili for just $9.50. Yes, support the sponsors. And as we mentioned before, the presentation on Wednesday is canceled, so we will see you Friday night at Amanhana, I believe. Thank you, and have a good night.